Okay, so we are on lab 5. So let me share my screen with the document, which are your lab exercises. Let me scroll up a little bit. Okay, so now we're starting the skeletal system. In this first lab, we generally look at a lot of the tissues, the large kind of regions of uh, long bones. We look at some different types of bones: long bones, short bones, regular bones. Um, and we look at some models of microscopic anatomy. And we also look at the region of bone elongation, which is called endochondral ossification. All right, so as usual, one of the first exercises, is to put some structure and functions together. So let's start with the anatomy of our long bones: the periosteum, endosteum, diaphysis, epiphysis, proximal or distal. The epiphyseal plate /line. Epiphyseal plate is an open growth plate. A line is a closed growth plate. The medullary cavity, red bone marrow, yellow bone marrow, articulating cartilage, compact bone, just be able to identify it. Spongy bone, just be able to identify it as the tissue, and then maybe, like with spongy bone, be able to identify a trabeculae versus, the red bone marrow portion. Okay, so we have down here our functions. So generally it's easier to grab the function and then match it to the structure that it corresponds with. So all produces all blood cells. A, that would be red bone marrow, right? So A is red bone marrow. Site of bone elongation, B, that would be our epiphyseal plate, epiphyseal line. Site of internal remodeling, C, that would go with your endosteum up here at the top. Then we have D, stores energy in adipose cells. That would be yellow bone marrow. E, ends of the bone where articulation occurs. Those are your proximal and distal epiphyses, because those are the knobby ends that we were talking about that in lecture that will have some hyaline cartilage functioning, or the articular ends of the bones composed of hyaline cartilage that reduces the friction between two bones that come together to form a joint. Then we have F, reduces friction when bones move. So that will be our articulating cartilage. As a structure, it's articulating cartilage. The tissue we will find there is called hyaline cartilage. Contains yellow bone marrow as an adult. That would be the medullary cavity. In children, we'd find red bone marrow there. H, bone region of longitudinal support. That would be the diaphysis. And then we have I, site of bone growth in diameter, also attachment site for tendons and ligaments. That would be the periosteum. All right, that would be the periosteum. All right, so we have something, a function to go with everything. And then we move on to the histology. So we'll look at those, in a few minutes when we go through the images, right? But when it comes to the histology, and I'll put these up when we get in the classroom again on the instructor's microscope. We're going to look at the compact bone and the spongy bone. And again, just like above, sometimes I just want you to identify it maybe on a model, but I don't necessarily, ask you to identify on everything, especially, in the slides, because the slides don't, the models, are, made of plastic and so forth, and the artist can put as much detail as you want. But when it comes to an actual slides, histologically, sometimes you can see these things, sometimes you can't. Right. So osteon, these are some of the specific components, or structures in compact bone, and then spongy bone, and we're going to add our functions to it, or a description perhaps. So let's identify all the structures for compact and spongy bone on models and slides, describe functions for all listed structures that we have here. So the osteon, that's the part of the compact bone, looks like a tree and all the rings of the tree. You know, the osteon is the repeating structural unit of compact bone because it repeats all over. Mhm. Osteocytes maintain bone matrix. Remember, we have osteocytes, osteoblasts, osteoclasts. Osteocytes maintain the bone matrix. They're found in compact bone region. Lamella, just identify on the model only. Lamella is just a word, another term for rings. So we have the osteon that's made up of rings of the matrix, you know, the hard stuff. And so, you know, lamella is just another word for a ring. A lacunae is a cavity, you know, area that houses an osteocyte. Canaliculi are narrow passageways connecting lacunae to the central canal, and therefore it allows diffusion of material through them. Nutrients will go from the arterial supply and diffuse out to the cells where the osteocytes sitting in their lacunae, and waste products and metabolic waste products will travel from the cells through the canaliculi to the venous blood and be evacuated away. The central canal is a large passageway in the center, a large diameter passageway for nerves, arteries, and veins. And yellow bone marrow, you know, we'll look at that, on the model and show you where it would be located. Also in a long bone, when we see a medullary cavity in an adult, we'd say that's where the yellow bone marrow would be found. Then when it comes to the spongy bone, it has a much different appearance. That's with the trabeculae that have these finger-like extensions of matrix, the hardish part of it, that look like the walls of a sponge. So the trabeculae are the hard components that add support. Osteocytes are the mature cells that maintain, you know, the trabeculae, the matrix found there, the hard components. The matrix, you know, I'm just going to show you on the models, are identified as the yellow stuff that you're, I guess, the hard portion of the bone that you're looking at that's arranged in rings. And then red bone marrow, which would be found between the walls of the trabeculae, you know, and red bone marrow produces all blood cells, all blood cells, red blood cells, white blood cells, platelets. Okay. Then we're going to look at what the cartilages look like on slides histologically, right? And we're going to see a few models where we'll be able to identify, oh, wow, that's a meniscus, you know, so that must be fibrocartilage, or this is the articular cartilage. It's light blue. It's hyaline cartilage. Most cartilage is going to be identified in the color of light blue in these models, and so forth. All right, so we need to be able to identify, right, the cartilage type. We need to identify the fibers found there and the cells. So the cells are easy. I would write those in first. Chondrocytes, chondrocytes, chondrocytes. Chondrocytes are cartilage cells, so they are common to hyaline, elastic, and fibrocartilage. So just write chondrocyte, chondrocyte, chondrocyte on the 3 lines, and then, you know, a comma. The fibers in hyaline cartilage, you know, I would just put none visible. You know, there's technically some fibers, but you know, you don't need to know the fibers there because they're really not visible to identify. And locations, well, hyaline cartilage is the most abundant. So you could write costal cartilage, the articular ends of bone. Costal cartilage is where cartilage attaches ribs to your sternum. The articular ends of bones, like we said. Your trachea, you know, areas in your trachea, your larynx, which is where your voice box is. The epiphyseal plates, where we have bone elongation. That's why it'll help you identify those areas, because it'll have that same soap bubbly appearance that hyaline cartilage has. All right, then we go to elastic cartilage, chondrocytes, as we said before. Elastic or elastin fibers. Right. Locations, all the EEE things. Ear, epiglottis, eustachian tube. Ear, epiglottis, eustachian tube. All right, and then fibrocartilage, again, chondrocytes. Fibers are collagen fibers. Locations, areas of a lot of compression. So we have the menisci of the knees. We have the intervertebral discs, these discs found between vertebrae. And then we have the pubic symphysis, not synthesis like dehydration synthesis, but symphysis with an M, like a symphony. And that's where the two pubic bones come together. Although you might not recognize some of these locations now, you will by the time we look at more bones of the axial and appendicular skeletons, it'll come together a little bit more. Okay, then when it comes to endochondral ossification, this is that epiphyseal plate, right? This is between the epiphysis and the diaphysis at the proximal end and the epiphysis and diaphysis at the distal end. And this is where we can take one long bone and lengthen it at both ends at the same time. So it grows very, very quickly, or rather quickly. And from the epiphysis toward the diaphysis, these zones are in the same sequence all the time. From the ends traveling toward, the middle, toward the diaphysis, we have a zone of resting cartilage that will be composed of hyaline cartilage. So you're going to see chondrocytes in there, lacunae, still bubbly appearance. Then you have a zone of proliferation where these cartilage cells start to replicate very rapidly and they stack on themselves. That was called a stacked coin appearance. So instead of being spread out and bubbly and so bubbly looking, they're going to be like you're looking at about 4 to 8 nickels or quarters stacked on themselves. So you're looking at them sideways. You can see these cells. And as these cells start to die off, these chondrocytes, they start to go from being stacked and kind of flat, to being puffed up and round. I call them the chubba lubba cells. They start to take in, as they start to die off, more cytoplasm. So instead of looking at them sideways and flat like a pancake, they're going to be big and round, where you can see a nucleus inside and then all the cytoplasm. So they have a very large translucent appearance. Right? And these are, in layers that we're going to look at. Now, they might be right on the horizontal. They might be a little oblique, but they're going to be in layers in the same sequence all the time. All right. So you go from the hypertrophic, which means increase in size of cells. Proliferation means increase in the number of cells, to your calcified matrix area of, ossification. So where we're going from having hyaline cartilage and replacing it with spongy bone and bone components. So the calcified matrix area is going to look a lot like spongy bone, where you're going to see trabeculae and often red, greeny, pinkish, dark pink staining goo found in between the walls of the trabeculae. That's the red bone marrow. And that tells you you are in this calcified matrix, area of ossification, going from cartilage to bone. And then the last exercise we're looking at is blood calcium homeostasis. So how do we regulate blood calcium regulation? And just like many different what's called analytes, either ions, electrolytes, or substances like glucose found in blood, there is a normal range, right? And if you go too high or too low, you need to adjust it to bring it back into the normal range. And these are examples of negative feedback loops. You start with one scenario and you're going to end with the opposite. Remember, we said that negative feedback loops, you end with the opposite essentially of what you're starting with. So if we look at calcitonin, that's one of the first hormones we're going to look at, and then we have parathyroid hormone. These are the main hormones that regulate blood calcium levels. And we look at this again in AMP2. So the more you get this down now, the easier to, out of like the 13 hormones you have to know in AMP2 in the endocrine system, the better. Okay, so like virtually any negative feedback loop, we have a stimulus, just go below that stimulus, a production site, a target site, and an action / response. Technically, the action is really different from the response, but we found it's easier to just talk about the action and then what you're ending with. It makes it a little more consistent when we look at all these different hormones. Okay, so the hormone is calcitonin. Below that, we have a little dot with a phrase. These are the phrases or answers that go into these 3 blank lines. All right, so let's start with the stimulus. The stimulus is high blood calcium levels, or higher than normal blood calcium levels. Right? So the calcium levels are going higher than normal in blood. Right? What are we going to do? Right? So what is the production site of calcitonin? it would be the phrase here in the center, the parafollicular cells in thyroid gland. So you're going to take that and you're going to write that into the production site. The thyroid gland is found here in the neck area. You have different cells there. You have follicular and parafollicular cells, and those different cells produce different hormones. So it's good now to identify and write, and rehearse that name or the term parafollicular cells of the thyroid gland, because you won't get it confused, hopefully later, with follicular cells of the thyroid gland. Follicular cells produce what's called T3, T4, but the parafollicular cells produce calcitonin. All right, so the target site, now we've crossed off that option. The target site is going to be osteoclasts in bone. Remember, osteoclasts breakdown bone. And the action response is that last phrase. So often the action response is going to be a little bit lengthy because I said we kind of combine the action and the response together. But this is essentially what you're going to do. And there's a twofold here, actually, that you do. Right? You inhibit the osteoclasts, and you stimulate your osteoblasts to increase calcium deposition and thereby decrease the blood calcium levels. So let me go back. So what's calcitonin going to do? All right, you have too much calcium in the blood. You don't want the osteoclast to keep breaking down bone and putting more calcium in there, right? Because that's just going to further elevate it. So you inhibit the osteoclasts. They're the ones with all the lysosomes, and you kind of put a muzzle on them so they can't do anything, right? And then the osteoblasts build up bone. Well, now that you have an excess of calcium, now's a good time to take all that calcium and store it in bone as much as possible and really strengthen those bones. And osteoblasts build up bone. So one cell you're going to put a muzzle on, the other one you're going to stimulate it to go, hey, now that there's all this extra calcium, let's store it in bone as much as possible, right? And whatever is excess calcium can just be eliminated through urine, out in urine from the kidneys. And we won't absorb we won't actively try to absorb calcium in foods that's passing through our digestive system. All right. So that ultimately lets that calcium level drop back down to the normal level. And now you stop secreting calcitonin. All right. So we start with higher than normal blood calcium levels. What's going to happen? The thyroid gland, or the parafollicular cells of the thyroid gland, are going to secrete calcitonin. Calcitonin inhibits the osteoclasts, stimulates the osteoblasts to store calcium, and thereby, right, we store some of that calcium, right, and now the blood calcium levels start to come down to a normal range. All right, but now we have the reverse that can happen. So now we look at parathyroid hormone, and parathyroid hormone comes from the parathyroid gland. Luckily, that gland only produces one hormone, so we call it the parathyroid hormone. All right, so now we have the negative feedback loop to the bottom. Now our stimulus is low blood calcium levels. Now they've gone lower than normal, really, blood calcium levels. All right. The production site is the parathyroid gland, the last option. Right. The targets, there are two targets. So parathyroid gland, how it works to elevate blood calcium levels is a little more complicated than just having an excess of something. All right, so parathyroid is going to do a couple things. It's going to target the kidney, all right? And at the kidney, all right, our action responses are going to be the function of the kidney once the parathyroid hormone targets it is going to convert vitamin D to calcitriol. Right? And calcitriol travels to the small intestine to increase calcium absorption there. It will also stimulate the kidneys to reabsorb as much calcium as possible from the urine that you're forming there. So there's actually more than one thing that really will take place there. But the parathyroid hormone also targets the osteoclasts, right? And what do we do with the osteoclasts? Well, now we want them to go to work. So we stimulate our osteoclasts. Right? And we let them break down some of that bone that we previously used to store calcium when it was in excess. Now we want to kind of take it back out of our savings bank and we want to put that calcium back into blood. So we stimulate the osteoclasts, let them go to work, use those lysosomes, break down some of that bone and let that calcium enter the bloodstream. And as a consequence of both of those things, all right, the conversion, what we do with the kidneys and how we stimulate the osteoclasts, ultimately elevate our blood calcium levels back to a normal range, increase our blood calcium levels. Calcium is required for cardiac muscle and skeletal muscle and smooth muscle contraction, so we have to maintain appropriate calcium levels. All right. So one, hormone is used when we have low blood calcium, we end up with higher blood calcium. The other, calcitonin, is used when we have higher normal blood calcium levels, and we end up with normal or decreased blood calcium levels. So they work in opposition to one another. All right, so that's it for that document. I'm going to stop the share. I'm going to close that out. And now I'm going to go to the images so we can identify those as much as possible. So let me go back into the course page. We're going to open up. Let me go back in the lab and open up the images. I'm coming. Go into exam. So now we're in lab material for practicals. Exam 2, practical information. And let me go to lab 5, which is what we're on, and those images. So let me move up a little bit. There we go. And now we're going to open up those images. So there are a bunch of different images here. And let me change my share so I can get in there. There we go. We start by looking at hyaline cartilage, and it's a pretty good picture of hyaline cartilage, right? This is actually from the trachea, but this region right in the center, you see how it has the light lavender background? It kind of looks like a soap bubbly appearance here, right? That is the region of hyaline cartilage. Those little bubbles, each one is a lacunae that has an a chondrocyte in the center, and an osteo got stuck in the bone. Right? So we have a chondrocyte sitting in its lacunae, and here they had the light lavender background where these kind of look like soap bubbles. And that light lavender area is the matrix that those chondrocytes maintain. There are fibers there, but don't worry about them. They're not visible. So you just have to identify that that is hyaline cartilage. What are the cells, chondrocytes, and no particular fibers. Or I can point to that and I could ask you, what are some locations? So you need to commit to memory, 2 or 3 that are the easiest for you to remember. Articular ends of bones, costal cartilage, epiphyseal plates, trachea, larynx. And then the next slide is generally a little card that kind of gives you a little illustration of what you're looking at. The chondrocyte sitting in its lacunae. Okay, then we have, oopsie doodle, I'm kind of going back up. I need to move this over to the side. That would be great. Then we go to elastic cartilage. Right? Right. So, here he gave you some of the locations for you to commit to memory. Tissue, if anybody asks you what the tissue is hyaline cartilage. That's the formal name. All right, so then we go on to this next slide. This is elastic cartilage, and it's magnified a little bit more so you can get a better idea of what these chondrocytes look like sitting in their lacunae. And look at all those dark purple fibers. This is what I call the Scooby-Doo eye appearance, is that those are elastic cartilage, elastic fibers or elastin fibers that take up stain. So it has a very dark purple contrast to those chondrocytes sitting in the lacunae. In hyaline cartilage, the matrix is rather pale staining. So, between that and the chondrocytes in the lacunae, they're both kind of pale looking, but now you start to see a stark contrast. And that's how you're like, ooh, that's got to be elastic cartilage, dark purple fibers, dark purple compared to the chondrocytes in the lacunae. Elastic cartilage. So we have the cells are chondrocytes. They're sitting in their lacunae. And we have locations, ear, epiglottis, eustachian tube. Right, ear, epiglottis, eustachian tube. All right, then we go to the last of the cartilages, which is fibrocartilage, and we looked at that in lecture. You know, the picture I have in lecture is actually very good for this. It looks a lot like this. We have these blue, pale blue staining wavy lines, and every once in a while you can see a bright magenta staining nucleus of a chondrocyte. So it's rather pretty. The light blue with the fuchsia nuclei. All right, this is fibrocartilage, collagen fibers waving, waving, waving like a Van Gogh painting. So we have the light blue collagen fibers. We have a chondrocyte sitting in its lacunae, same cells. And then we have locations, intervertebral disc, pubic symphysis, or the menisci of the knees. All right, then we have compact bone. So here we have one osteon. You can see these rings. So the rings are called lamella. We can see these extra blackened looking oval slit kind of areas here that are found actually between lamella to lamella. That would be a lacunae. And in life, if this was live tissue, you would find an osteocyte sitting in that lacunae. But right now, it's just a cavity. It's just a lacunae. And then in the center, you have a big hole, you know, that's blackish also. But this is where we find the arteries, veins, and nerves. You can see these light lines that are radiating like spokes from a wheel out from the central canal to the lacunae. And those are the canaliculi, or can I lick your eye? Canaliculi that are narrow diameter passageways for diffusion of material to go from the blood supply, you know, in the central canal out to the osteocytes in the lacunae. And they all connect lacunae to lacunae to lacunae. Remember that even though this bone tissue, the matrix portion is very dense, is that it's actually live tissue. I mean, you have cells, live cells that need oxygen and nutrients and things, you know, to maintain that matrix. If you cut off the blood supply, you essentially smother those cells. They die, and you have what's called osteonecrosis. The bone starts to die off. All right. So here we have one osteon, which is the repeating structural unit of compact bone. Here we have the rings called lamella. Right. Then we have the central canal, which has nerves, arteries, veins. You know, there's some lymphatic structures in there as well, and NAV, called naval. Right. You have the little canaliculi, which are narrow hair, he calls them hair-like, but you know, they're very narrow diameter passageways for diffusion of material, you know, from the blood supply out to each of these cells that are, you know, at different distances, you know, from the central canal. So everybody's got to eat, you know, and that's what the blood supply is for, right? To allow diffusion of oxygen, nutrients, and waste material. Okay, compact bone. We had two major regions of the bone. We had the diaphysis, which adds support and protection, you know, a lot of linear vertical support. In compact bone, we had the diaphysis and we have the epiphyses, which are regions. Right? Here we have what's called like a large model of one osteon, right? Right. So here we have the big hole in the center, the central canal, which has, you know, arteries and veins, arteries in red, veins in blue, nerves in white. This here, sometimes they'll be identified in yellow. And if you see green things somewhere, that represents a lymphatic structure. It's the big hole in the center, right? The rings of matrix are called lamella, okay? The matrix is identified in that yellow color. All right, so we have lacunae, which are the cavities. No, I didn't know what he was really pointing to there. Hold on. Let me go back one. Let me just kind of tell you what everything is. All right. So the yellow that you see here is the hard part of the matrix. Right. It's arranged in rings that are referred to as lamella. That's better. All right. Between each lamella, right, you see these cavities here, the little indentations, kind of tear-shaped or looking. Those are the lacunae, right? So the lacunae is a space, right? In life, right, when it's live tissue, you see these blue nuclei, right? Those are the osteocytes that sit in that lacunae, right? And these red lines here represent, you know, some of the cytoplasm of that cell that's in those canaliculi. Can I lick your eye? So the little fine grooves here, right, that are radiating from the lacunae, extending like finger-like extensions toward the central canal, those are the canaliculi, right? They're just empty, all right? The red lines, we generally point to the red lines to identify that, because you can actually see the red. You can say, what is, where the red is there, what is that structure? Canaliculi, right? The empty oval there is a lacunae. And the blue, if someone points to the blue and says, identify that cell, quote unquote, then it must be the osteocyte. All right, but now we'll look at kind of each one. So central canal and lacunae. Okay, central canal and lacunae. All right, then we're pointing to the red stuff here. And so we're trying to identify specifically the canaliculi there. And one is pointing to the osteocyte, maintains bone matrix. Right? Canaliculi are small passageways for nutrients, waste material to the blood. Right? Then we have, the matrix and rings of matrix. I generally don't, ask you those, the lamella and, and the matrix. I generally don't ask you the matrix and the rings, and I generally don't ask you to point them out, on the model also, because you're never quite sure, is she asking me about the yellow stuff, or is she asking me about the ring? So I will say that if I do ask you a question, I'll ask like, what are these rings of matrix? And then it's lamella. If I ask you, what's that hard yellowy substance? Then it might be matrix. And that's how you distinguish them generally, distinguish between the two. But I don't ask that frequently. Okay, this is a picture of spongy bone. All right, so that was compact bone. Now we're on spongy bone. The pink orangey kind of material here that looks like little islands or finger-like extensions here, that is the trabeculae. The region between the trabeculae, which you can't see a whole lot of red greeny goo stuff, but you can see some adipocytes in there. All that would be the red bone marrow. When we look at endocardial ossification, and those different zones at the epiphyseal plate, that really I find is like the best example of spongy bone that is more consistent with pictures that you're going to see in your textbook and things, or in the lab book if you actually have that. Okay, so the pink part, the little finger-like extensions here, that is the trabeculae and the red bone marrow inside. Function of the red bone marrow produces all blood cells. The trabeculae, lends architectural support. Something like that is fine. Okay, so he made you a little card. So the finger-like projections here, this is the trabeculae. It kind of has a lattice fence kind of look to it. That's where we would find the osteocytes. And then in between those, in another subsequent picture, I'll show you that it has more of a red grainy or dark pink grainy goo, grainy appearance with some adipocytes in their fat cells. Then you're in red bone marrow. It produces all blood cells. Okay. Spongy bone is located in the epiphyses and in the, right now it's just going to be the epiphyses, and I won't ask you a function of spongy bone. I'll just ask you the structure, trabeculae, and the red bone marrow and their functions. Okay, so here he has them in a different sequence, but this region here, this is a long bone that's kind of on an angle, right? It's kind of on a 45, not quite 45-degree angle. This end to the upper right-hand corner is the epiphysis, all right? So the knobby end of the bone. And this to the lower left-hand corner is part of the diaphysis. So where that knobby part of the epiphysis meets the shaft of the diaphysis is this region called the epiphyseal plate, which implies that we have an open growth plate there where the bone can elongate, right? And from the epiphyseal and upper right-hand corner toward the lower left-hand corner are these zones, and always in the same sequence, all right? If this was flipped around, it would still go in the same sequence from epiphysis end toward the diaphysis. So we have our zone of resting cartilage, which means this is this area here that looks just like hyaline cartilage. And it's hard to see right now, but you'll get the knack of this, especially if you can come to class, all right, and I can zoom that and I can get it on the camera, well, you'll be able to see the difference between these after a while. They look similar, but then you're like, oh, now I can see this, So this region is your zone of resting cartilage. Then as we travel down and to the left toward the diaphysis, the zone where we have these cells stacked up on one another, right? And they're going to be kind of in this 45-degree angle, rather linear, is our zone of hypertrophy, which means cells increasing in number. Then as we go deeper, we get to this layer where the cells are hypertrophying, which means those same cells are now going to increase in size. And they start to blow up, like I call them the chubba-lubba cells, because all of a sudden they're really big. You can see all this cytoplasm with the nucleus in the center. All right. And then as they die off, they get replaced down here to the lower left-hand side by your zone of calcification, ossification. This, you can see here on the lower left-hand side, this is beautiful red bone marrow. You can see light pink regions, which would be the trabeculae, the little finger-like extensions, the lattice work. And then you can see this red greeny goo in between those. That's the red bone marrow. And now it looks actually red. Okay, so this is endochondral ossification. It occurs, between the epiphysis and the diaphysis. Technically, it's called the metaphysis. All right. So here, the arrow or the pointer here, I think it's just some kind of a stick or something that he's using, maybe, is pointing to your zone of calcification, ossification, and then from there to the left, is that the completion of that zone. That should be that, or I really wish I could magnify this a little bit more. He's right at the border of hypertrophy and calcification. I would point more to that red greeny goo area, right? Because he's going in different sequence. This, he's definitely in your zone of resting cartilage. About halfway down, go to the left of that, and you can see these dark cells that are kind of stacking on one another. I don't know if you can see my little cursor arrow, but I'm pointing to those all here. That is your zone of hypertrophy. So he's pointing to resting cartilage. Next to that would be hypertrophy. Further, where you can see these cells, you can almost see a little nuclei in some of these big, soap bubbly chubba-lubba cells here. That would be a zone of hypertrophy. And then your zone of calcification. So we have resting cartilage. Oh, I called that, I'm sorry, that's proliferation where the cells are stacked on each other. So yeah, I'm getting distracted again. And then we have hypertrophy and zone of calcification, ossification with all that red bone marrow and trabecula you can see there. So I think what I would have done if I was him is put these in one particular sequence or another. Maybe I'll try to move those around. It says that they're numbered. So if I take them out of the numbered range, then you're wondering why I took them out of the numbered range. But this is a little magnified. You can see a little bit better, right? Same orientation. Upper right-hand corner is your epiphysis. So that's where we find all this hyaline cartilage. That's your zone of resting cartilage. Then next to that, like now that it's magnified a little bit, do you see these stacks of cells? Because they look kind of like brownish, where they're stacked on each other. Sometimes it's just 2, sometimes it's about 4 or 5. So that would be your zone of proliferation, now that I said that correctly. Then to the left of that, you can see these clear cells. That region is the cells that were just proliferating. Now they're getting hypertrophied and they're expanding and swelling. You see a lot of cytoplasm, these big cells with a nucleus in the center. And then they die off and get replaced by the tissue to the lower left-hand side. That is your zone of calcification, maybe called ossification. And now we're replacing that cartilage with all this spongy bone. And you can see trabeculae and the red greeny goo, which is the red bone marrow. All right, so sorry I said that incorrectly before. And I think he gives you a little card that puts these, in kind of sequence. All right, so this process that's occurring there is called endochondral ossification, right? Within kind of cartilage and ending up with bony stuff. That's essentially what it means, right? You have a zones of resting cartilage, zone of proliferation, increasing in number, stacked coin appearance in this order, right? Zone of hypertrophy, cells getting big, and then followed by a zone of calcification. If you write backslash ossification, that's fine too. Now we've replaced that cartilage, hyaline cartilage, with some kind of spongy bone. Okay, so it's a funny, it looks like a peanut, doesn't it? But it's trying to show you the two ends of the epiphyseal, the epiphyseal ends with the diaphysis in the middle. All right, so from the epiphysis end, regardless of whether it's, going right to left or left to right, you start at the epiphyses, and that's where you find the hyaline cartilage. So that's your zone of resting cartilage. And then traveling toward the diaphysis, right? Then you had your zone of resting cartilage to the zone of proliferation, to the zone of hypertrophy, to the zone of calcification, ossification. And you start with hyaline cartilage, and then you end up with spongy bone. Okay. Always in that sequence. They'll never get rearranged. So, start at one end of the cell, start at the epiphysis and work toward the diaphysis, or start, if it's easier for you to identify the spongy bone area, then go from there toward the epiphysis. That would be your zone of, calcification. And then, you would have your zone of hypertrophy to zone of proliferation to zone of resting cartilage. We'll always go in the same sequence, either right to left or left to right. And we'll look at that more again in lab. Okay, these are examples of the different shapes of bones. This is an example of a long bone. You don't know what that is, but it is a femur, a femur. That's your thigh bone. Right? Example of a long bone. It's really, really long. Then we have, this is what I call the pie wedge of compact bone model. And it's really rather large in the lab, so it serves as a good one. You had that just that single osteon. It was a great big osteon. This identifies a lot of those same things, but it's a bit smaller, so that one big osteon is much easier to identify your canaliculi and lacunae with that. This helps you identify the cells, the osteocytes. It lets you identify the endosteum and periosteums. You can see the osteons, the repeating structure of the osteons. You can see a really nice central canal. They're just slightly different colors. Right. So here we have one on the outer gray connective tissue to this region of compact bone, this rim of compact bone. So that would be the periosteum. Right? This is where you find osteoblasts and osteoclasts for bone kind of remodeling. You can increase the width of it and then repair it in the case that you had, let's say, a fracture. Periosteum. All right, then we have the inner gray layer. So this is kind of, I would call like a piece of pizza, a pie wedge kind of a shape to it. So the outer gray is the periosteum. This inner gray layer represents the endosteum. Endosteum means within. Right? Another area of growth of thickness, or maybe remodeling a bone when it's been damaged. All right. Here we have a live bone. It's a real bone. Butterflied open, so you can see this is the epiphysis and this is the diaphysis. And here you can see this network. It kind of looks like little finger-like projections of hardness, kind of like a sponge, the walls of a sponge, or maybe, lattice-like structure. All right, so that would be, this region is the epiphysis. The one is the diaphysis. In the epiphysis, we have spongy bone, right? That's the tissue we find there. Right. And those hard components that you're looking at here, all right, traveling in different directions, is the trabeculae of spongy bone. So the tissue is called spongy bone. That structure is called trabeculae. And in life, because this is dead and everything's evaporated away, between the walls of that trabeculae, you would find red bone marrow. And that's where all blood cells come from, are made. When you look at the diaphysis, you see this concave region there. That concave region running all the way down the length of that long bone is the medullary cavity. In adults, we find yellow bone marrow there. In children, we would find red bone marrow there. Right, yellow bone, so number one is the medullary cavity. That region of the bone is called the diaphysis, the shaft of the bone, right? Triglyceride storage, anything that goes with fat, stores energy, cushion and insulate organs, whatever you used in the lipid functional category in chemistry lab, is fine by me. And at the epiphysis, we find the spongy bone, I mentioned specifically the structures you would find there because you should know that, the trabeculae and the red bone marrow. Okay, this is that same bone kind of closed up, and it's just pointing to those regions, epiphysis versus diaphysis. All right, epiphysis versus diaphysis. And here, this is where he's just magnified a little bit more, where you need to identify the trabeculae, and the red bone marrow. This is also that junction. And, I think I would use a better picture, but I will just tell you is that the card that follows this isn't necessarily, and explaining what you're kind of quite looking at here. We believe that one little line across here is the epiphyseal plate. And now that it's closed down, it's called an epiphyseal line. It's replaced by a solid piece of bone. Yeah. But you'd have to really look at the question and read the question, because that's not very easy to pick up on if you were looking at that. You don't know, they asking about a trabeculae, are you asking about red bone marrow? Are you asking about the epiphysis? So I would probably not use that. I would use a previous picture and then say, what's that structure pointing to? What's the tissue? Spongy bone. What's that structure that you can see that's hard? The lattice-like structure, that would be trabeculae. What might be the function of that trabeculae? What would I find between those walls of the trabeculae that I'm looking at? Red bone marrow. What would be the function? Produce all blood cells. See, it's kind of tricky to see, but that is what's called an epiphyseal line. When it had all the hyaline cartilage there, then it would have been called an epiphyseal plate. I don't think I'm going to ask you something like that. If I gave you, there's other bones that we could use in the lab. There'll be a big long bone, like a femur, that's actually a model, so it's plastic. And we put these blue lines to the outside surface between the epiphysis and the diaphysis. I mean, that's a really nice example, but I'd use a different way of demonstrating it so that you see this very dark, kind of like the blue on this joint model here. That's from a knee. That dark blue, you'd have this line that's been kind of painted on there, and so that's to identify the epiphyseal line, and or the epiphyseal plate, really, because blue generally indicates hyaline cartilage. The region of the bone above that would be the epiphysis. The region, below that in the shaft would be the diaphysis. But that line, that blue line would represent endochondral ossification as a process there, and that's your epiphyseal plate. But that's why these images that are in here, uploaded to the course page like this, aren't the only things you're responsible for. You're responsible for things in the lab, and that's why we're trying to use some of those as well, and to zoom it and use the camera to get some of those, so you can get a better idea of what we're talking about. All right, this happens to be a knee joint. We'll look at it now. We'll look at it later also when we look at joints. All right, but here we have two bones that come together. This is a femur, this is a tibia, right? But we'll get to that in the appendicular skeleton. The blue at the end of that bone, because that region above that is an epiphysis of the bone, all right? The blue is the articular cartilage at the end of the bone. So as a structure, that number one is articular cartilage. If I ask you the tissue, it's got to be either a CME or N, a connective muscle epithelial or nervous tissue. And it's blue, so you go, ooh, that's hyaline cartilage, because cartilages are considered tissues, all right? As a structure, I can't tell you How many times I have to reiterate this? As a structure, that's articular cartilage, but the tissue that makes that structure or forms that structure is hyaline cartilage in blue. All right. Then below that, you see those white little discs there, those pads. Oh, those are menisci of the knees. All right. And those are formed of what tissue? Fibrocartilage. So pointing to two, you go, okay, what's that structure? It's a meniscus or a menisci, or, on the knees. That's fine. What tissue? Fibrocartilage. Okay. What cells are we finding in either one of those? Well, it's all cartilage, so it's got to be a chondrocyte. Maybe, what fibers would I find in number two? Well, once you know it's fibrocartilage, then you know it's collagen fibers. Okay. So the nice thing about this model also is that it shows you in the epiphyses, the yellow versus the red stuff in there. That's trying to tell you that that is spongy bone. So the yellowy parts, vanilla, maybe color parts, would represent the trabeculae, and that reddish stuff like rain gritty goo would be red bone marrow. I don't think we use that right now for this. I don't think anybody does. We will come back and look at these green structures on the side. Those represent ligaments and tendons. We'll look at those again when we use this as an example of a synovial joint when it comes to types of joints. Okay, so one is the articular cartilage. All right, it's composed of hyaline cartilage. That articular cartilage helps to reduce friction and acts as a shock absorber. So does the meniscus. And the meniscus is composed of fibrocartilage. All right, so even though it doesn't tell you here, it just says the structure, you need to know what tissues are found there. See, and then here he points to that spongy bone. I don't use that at this point. I don't think there's a question like that. All right. I generally use those slides, honestly. Okay, so that's it. That's all the images. Whew. I'm gonna stop sharing that. Okay, so so that's the lab. I know you guys just took the exam and you're not too excited about going forward, but I would start at least what I call that passive studying, is just looking at pictures and looking at some of these things. Take out your textbook and look at something. Take out, use a highlighter and highlight, the structures that you have to know for lecture and lab, in your textbook maybe, and start looking at those things, because, the more you look at it, the more it starts to become familiar and part of your vocabulary. There's a lot of terminology starts sounding the same. And when we get to the muscle system, it's the same way. They're microanatomy. You have a sarcoplastic reticulum, sarcolemma, you have a lot of terms that all start sounding the same. So the sooner you start practicing that and reviewing it and over and over again, I think the better off you're going to be and the less confused you'll be. So, all right, then I will see you in class next Thursday. If you want to come to class, come. If you want to stay home and Zoom it, you can. If you want to take turns, kind of whenever you go back and forth, that's fine. I think we'll just opt for is the first 12 people to come in lab and do it there. These are good ones to kind of see face to face, but please review this and fill in your, the lessons there so that you've reviewed it before you come in. And that way, you might only have to be there a half an hour, 40 minutes or so. The quiz on this material will be the following, Wednesday night, because your quiz is the night before lab. So, all right, see you next week. Bye.